Hi, my name is Kate Byrne. I'm an assistant professor at OHSU. I've just started my lab there. My work has focused on generating T cell responses against pancreatic cancer, which is what I'm going to discuss today. So, pancreatic cancer is the third leading cause of cancer related mortality in the United States. It's higher than breast cancer. And unfortunately, this disease is highly treatment resistant, not just to common treatments that have been used for a number of years, but also importantly, it's very resistant to immunotherapy. There's a number of reasons for this, and we don't fully understand the tumor microenvironment and how to overcome these barriers. The pancreatic tumor microenvironment has a low average tumor mutation burden, but it's not zero, and there are a fair number of patients that actually have a moderate number of T cells that infiltrate into the tumor site. And we do know that for those patients that have a higher T cell infiltration, they do have a higher survival rate. So it does suggest that there should be some potential to harness the immune response against this tumor. However, even patients that have a high tumor mutation burden that are MSI high have a very low response rate to immune checkpoint therapies. So almost no patients are responding to immune checkpoint blockade. Here I'm just showing patient samples in the tumor microenvironment, and you can see that there's a very high CD45 infiltrate into the tumor site, but most of these cells are usually myeloid cells, and for the majority of patients, there's actually relatively few T cells in the tumor site. So to understand the barriers to developing immune responses against this tumor, in the lab, we're taking advantage of the KBC mouse model. These mice have mutated KRAS and loss of P53, specifically in the pancreas. These are the two mutations most commonly found in patient samples. And you can see here that if we look at the histology, the tumors in the KPC mice really recapitulate the tumors in patients. So there's a high CD45 infiltrate into the tumor site, but again, most of these cells are myeloid cells. And especially in the mouse model, you can see that there's a true dearth of T cells in the tumor microenvironment. We and others have described multiple mechanisms of immunosuppression in the pancreatic tumor site, including stromal elements and tumor cell intrinsic features that really prevent T cell infiltration into the tumor and provide a very strong barrier to immune responses against this tumor. Similar to patients that are the hardest to treat, the KPC mouse model has a very low tumor mutation burden. There are very few T cells infiltrating, as I'm showing here, and they're highly resistant to therapeutic intervention. Here, we are just showing some examples of the therapies that these tumors are resistant to. We used the KBC mouse model to generate a tumor cell line that we could then implant back into wild-type mice. And you can see here, when we treat mice with either gemcitabine alone, nab-paclitaxel alone, or the combination of the two, which is a standard of care for patients with pancreatic cancer, we have no impact on the tumor growth rate in these mice. Similarly, if we treat the mice with checkpoint blockade, either anti-PD-1 alone, anti-CTLA-4 alone, or even the combination of the two, we really cannot control this primary tumor growth. So the KPC tumors are highly resistant to standard of care chemotherapy and immune checkpoint blockade, modeling, again, those patients that are the hardest to treat in the clinical setting. So just to orient us to how we're thinking about generating an immune response against this tumor, I wanted to start with the tumor immunity cycle. Generally, you can imagine an immune response against a virus or another pathogen will have multiple steps along the path, including antigen presentation, T cell priming, infiltration of the T cells into the target site, and then killing of the target cells by the T cells. In the tumor immunity cycle, we know that one or more of these steps are broken somewhere along the path, often several. And especially with regard to current therapies, we know that most of the treatments are focusing on this side of the effector response against a tumor. There's a presupposition of an existing T cell response that needs to be reinvigorated or rescued. And immune checkpoint blockade has done that very well in many aggressive tumor models, such as melanoma and lung adenocarcinoma. In those cases, blocking inhibitory receptors, such as anti-PD-1, CTLA-4, or now also TIM-3, allows for T cells to infiltrate back into the tumor site or recover their function within the tumor site, leading to the death of cancer cells. However, we know that in many tumors, such as in pancreatic cancer, this is simply not working. And so we need to think about other ways to generate this T cell response against a tumor. Our lab has focused on the hypothesis that priming a T cell response may actually overcome the therapeutic resistance that we're seeing in pancreatic cancer and many other aggressive tumor types. So we've decided to focus on the step of antigen presentation and the priming and activation of T cells against the tumor. To do this, we're taking advantage of the agonistic CD40 antibody. This antibody mimics the natural CD40 ligand, and when we administer it, we have cross-linking of CD40 on dendritic cells or other antigen-presenting cells, driving the licensing and maturation of these APCs. As a result, we have the generation of a T cell response that expands, and we've shown that this T cell response actually can traffic into the tumor site and mediate tumor aggression. So we had two key questions that I wanted to address in this presentation. We first wanted to understand if therapeutically enhancing T cell priming would actually elicit anti-tumor immune responses against pancreatic cancer. And if that is true, we then wanted to determine what the correlates of response to immunotherapy were, especially within the tumor microenvironment. So as I described before, we made a cell line from our KPC mice. We could then implant that cell line back into B6 mice. And here I'm just showing our treatment schema where we implant our tumors around 2 weeks before we begin therapy, waiting until we have an established tumor. At that point, we can treat mice, as I showed before, with gemcitabine and nab-paclitaxel, standard of care chemotherapy for patients with pancreatic cancer. 2 days later, we followed this with a single dose of agonistic CD40, and then we monitored these mice for survival. So here I'm showing the survival curves of mice treated with our combination of agonistic CD40 and gemcitabine nab-paclitaxel, shown here in the open circles. And you can see that mice that received this treatment regimen actually survived long-term compared to mice treated either with chemotherapy alone, CD40 alone, or vehicle controls. We did a number of studies that I'm summarizing here in this slide to show that we actually had clonal expansion of our T cells locally within the tumor microenvironment. And we also found that those cells had a higher functional quality. There were more cytokine production and activation of T cells in the tumor microenvironment after we gave this combination therapy. Importantly, we assessed the role of innate immune sensing pathways, such as type 1 interferons, the TLR pathways, including MyD88 and other TLR signaling pathways, and STING and other innate immune sensing molecules inside the tumor microenvironment. Contrary to what was published at the time, we found that none of these innate immune sensing pathways were required for the generation of this T cell response, showing that CD40 was really able to drive a unique T cell response in the tumor microenvironment. We also found, as we predicted, that BATF3 dendritic cells were required for CD40 efficacy, as was interferon gamma. Importantly, we found that when we gave agonistic CD40, we drove a CD8 infiltration into the tumor site. Here I'm showing a control tumor, a mouse from the control group that didn't receive any therapy. There's no T cell infiltration into the tumor site. But when we looked after therapy, we could see a robust infiltration of T cells into the tumor. Knowing that we had driven T cells into the tumor site, we hypothesized that if we now added in immune checkpoint blockade, because we had that pool of effector T cells, we may actually see an improved immune response using agonistic CD40 with chemotherapy and dual immune checkpoint blockade. So here's the treatment schema that I showed you before, and you can see that we have, as I showed you before, our response with chemotherapy and CD40. And now we did the same treatment schema, except we added in anti-PD-1 and anti-CTLA-4 together, ICB, immune checkpoint blockade, concurrent with our treatment with chemotherapy and agonistic CD40. And we gave multiple doses of our immune checkpoint therapy. When we did this, you can see now in the purple line that the mice responded very well to this combination agonistic CD40 and immune checkpoint blockade with our chemotherapy. In the green line is similar to what I'm showing you on the other side of the slide, our CD40 with chemotherapy, and you can see survival is extended, but it's not as striking as when we're giving this combination with immune checkpoint therapies. We wanted to translate some of these findings into the clinical setting, and so we partnered with Stand Up to Cancer, CITN, PanCan, and Roche to do a phase 1 neoadjuvant adjuvant trial. So in this clinical trial, patients were segregated either into arm 1, which got anti-CD40 or salukelumab alone in the neoadjuvant setting, or arm 2, where they got a combination of gemcitabine and nab-paclitaxel with salukelumab, which is agonistic CD40. And patients received a single dose of agonistic CD40 with or without the addition of the chemotherapy, followed by tumor resection at day 15. And then they underwent recovery, and then all patients underwent adjuvant chemotherapy and agonistic CD40 after surgery. And we were able to analyze a number of different metrics, but importantly for this talk, I'm going to focus on the biological correlates specifically within the tumor microenvironment to understand the impact of using agonistic CD40 in patient tumors. So in order to do this, we partnered with Lisa Cousins at OHSU to use her multiplex IHC platform to understand what's happening really within the tumor microenvironment. So here, Lisa's lab has developed a multiplex system where she can stain for a number of different markers within the tumor microenvironment in an iterative staining process. And then in that single slide, we can assess where each of these different markers is expressed. And you can see here, by using this detailed panel of immune markers, we're able to identify a number of different immune and tumor microenvironment cell populations, as well as several phenotypes, as well as cell functions on these cell populations. And we can do this in a very detailed manner across a number of different samples. And this will allow us to understand what is happening in the tumor microenvironment after we're giving agonistic CD40. So importantly, in this study, we did not have a baseline biopsy from these tumors. So instead, we took advantage of Lisa's group creating a pancreatic tumor atlas where they had a large population of patients that received either no treatment before tumor resection or chemoradiation. And we were able to use the exact same panel in those patients and then compare it to the patients that received agonistic CD40 to understand if there were any differences in our untreated tumors versus those that got agonistic CD40. So here I'm showing major cell populations that we identified in the tumor microenvironment on the y-axis, and then our different patient samples are shown here on the x-axis. And this is done by an unsupervised clustering. We found that the tumor microenvironments clustered into three main phenotypes: a granulocytic-rich microenvironment, a T-cell-rich microenvironment, and a myeloid-rich tumor microenvironment. Importantly, you can see that here in the black bars are patients that received agonistic CD40. And you can see the majority of them actually cluster into this T-cell-rich tumor microenvironment cohort, meaning that patients that were treated with agonistic CD40, again, which is called selukelumab, had an 82% chance of falling into this T-cell-rich phenotype. Whereas patients that received either no treatment or received only chemotherapy or chemoradiation therapy in the neoadjuvant setting had only between a 23 and a 37% chance of falling into that T-cell rich category. So again, 9 out of our 11 patients that received agonistic CD40 ended up having a phenotype in the tumor microenvironment that appeared to be enriched for T cells, really suggesting that CD40 was altering the tumor microenvironment in favor of T cell infiltration into the tumor site. We were further able to dig deeper into this data by taking advantage of Lisa's system to break the tumor into a number of different regions. Using pathological annotations that identified the tumor and the tumor border, the team segregated the tumors into 4 different parts, across the tissue slide. So there's the intratumoral, which is really the tumor nest, where the tumor cells are all located, followed by a border region that's slightly farther away from this edge of the tumor. Then we have a spanning region that covers 50-50, the border region, and then this distal region. And this allows us to really understand what's happening across the entire tumor microenvironment and the adjacent tissue that should be separate from what's actually happening in the tumor site, theoretically. This allows us to really understand the difference between what's happening right inside the tumor nest and across the entire tissue type. Importantly here, we're showing different cell populations in different colors. You can see here on the y-axis, it's showing which cell population is being shown. So for example, up top, in these patients from our treatment naive group, you can see the B cell population is shown in orange, followed by our CD8 T cell population in red, and then the CD4 population is shown here in blue. And you can see also the relative density of those cells by showing how thick the line is. So you can see B cells are highly present, for example, at the border of the tumor and inside of the tumor nest, followed by CD8s being highly present in those tumor sites, whereas CD8s are more abundant when we're looking also in this spanning region. So away from the exact border and tumor microenvironment, CD8s become more enriched in these treatment-naive patients. So by doing this, you can kind of imagine the differences in the densities across the entire tumor microenvironment. And again, we can compare these trends in treatment-naive patients to patients that received agonistic CD40. So here I'm showing our treatment-naive patients, followed by our patients that received chemotherapy or chemoradiation in the neoadjuvant setting. And you can see that there are some similarities between the two groups. Notably, B cells are a highly abundant population in these two patient cohorts. On the right side of the slide, we have our patients that received agonistic CD40 in the neoadjuvant setting, either with or without the addition of chemotherapy. And here, importantly, you can see that T cells have now become the most abundant immune cell population present from the distal site in the slide all the way into the intratumoral region. Both CD4s and CD8s have become a highly abundant cell population that's present within the tumor microenvironment. So you can see that both CD4s, shown in the light blue, and CD8s are present in increasing frequencies and remain the dominant population within the tumor microenvironment itself. So CD40 is not only increasing T cell infiltration into the tumor site, but it's also changing the spatial distribution of immune cells all across the tumor microenvironment within the tumor nest and distal to the tumor as well. We also looked in the peripheral blood of these patients. We wanted to understand if there was systemic immune activation occurring, as we might expect from giving agonistic CD40. So here I'm showing on the left side of the slide our CD4 effector cell population, and on the right side our CD8 T cell effector population. We looked at a number of different activation and potential exhaustion markers, and we found that almost all of these were increased in CD4s and CD8s. We also saw in CD8 specifically that there's an increase in Ki67, suggesting an increase in proliferation of these T cells. Instead of viewing this as exhaustion, we actually took this to mean that we were getting this priming of a T cell response systemically, and that those T cells were likely then infiltrating into the tumor site. And I will just mention that this peripheral blood sample was taken at day 8 after agonistic CD40. So this is halfway in between the treatment with CD40 and when the patient went in for tumor resection. So it's about when we would expect to see an increase in T cell activation in the periphery after treatment with agonistic CD40 that should drive T cell priming. So just to conclude on this first part, we found that actually pancreatic cancer is sensitive to immune-mediated destruction in the KPC mouse model. We found that both CD4s and CD8s are mediating this destruction, and importantly, the agonistic CD40 was able to bypass innate immune sensor stimulation. And this suggests to us that actually agonistic CD40 could be paired with other partners, but also that it's using a non-canonical pathway to drive this activation, which opens the opportunity to use CD40 where other immunotherapies have failed. We also found that CD40 agonism was altering T cell infiltration into the tumor microenvironment, both in mice and in patients, and that CD40 was driving systemic T cell activation. So when we think back to our key questions, we determined that yes, actually therapeutically enhancing T cell priming was driving tumor immunity, and we were able to identify a number of different factors that correlated with response to this treatment. This actually led to another question. As you may have noticed, there were some slight differences in the patients, whether or not they had CD40 plus chemotherapy. And around this time, I had a master's student joining my group who was very interested in understanding what the potential impact of chemotherapy was on the developing immunotherapy-induced response. In some cases, we know that chemotherapy can drive immunogenic tumor cell death and could actually enhance T cell responses against tumors. But in other cases, we know that chemotherapy can actually drive the death of the immune cells themselves because they are proliferating after immune intervention. And we didn't know exactly what was happening here. So to address this question, we set up a study similar to what I showed you before, where we gave mice agonistic CD40 and dual immune checkpoint blockade with chemotherapy, except at this time we also set up an identical cohort of mice that just received agonistic CD40 and immune checkpoint blockade, no chemotherapy. So here I'm showing you a waterfall plot where each bar is an individual mouse, and you can see that in the primary tumor response, the waterfall plots between our chemotherapy CD40 ICB treated mice were almost identical to mice that did not receive chemotherapy, and tumor growth control also looked nearly identical. But importantly, when we followed these mice long term, we found that the mice that received agonistic CD40 and immune checkpoint blockade with the addition of chemotherapy actually slowly succumbed to tumor burden over time. So these tumors eventually grew out, and it suggested to us that the quality of the T cell response in these mice that had chemotherapy was actually diminished compared to the mice that did not receive any chemotherapy. But we were also surprised to see that we actually had a really good response against the tumor in the absence of chemotherapy. And it suggested to us also that CD40 and immune checkpoint therapy was potent enough to drive a very robust T cell response against a tumor. So we wanted to interrogate this a little bit further. So we first wanted to understand what was required to generate this immune response against the tumor. Did you need both immune checkpoint blockades or just one? So here I'm showing the subcutaneous tumor model that we use, where we implant our KPC cell line under the skin of the mice. The barrier here to generating a T cell response is actually somewhat lower than in other sites on the mice. And you can see here that actually the immune response is pretty good if you use agonistic CD40 with either PD-1 or CTLA-4 or both. However, when we did this in the more aggressive orthotopic model, where we implant the tumor cells back into the pancreas itself, here it was very clear that you needed to have both anti-PD-1 and anti-CTLA-4 on board in order to get good tumor control. We also were interested in understanding the sequencing. We had used previous sequencing models to predict that we needed CD40 after chemotherapy. And so here we modeled giving agonistic CD40 first, and then giving immune checkpoint blockade instead of our traditional route where we gave immune checkpoint blockade first and then agonistic CD40. And you can see that the original plan, where we gave anti-PD-1 and CTLA-4 followed 3 days later by agonistic CD40, actually did slightly better than when we gave CD40 first. And it suggested to us that there may be some impact of combining CD40 and immune checkpoint blockade during the priming phase. We needed to have immune checkpoint blockade on board at the same time. So we next went to look at the induced immune response to determine if both CD4s and CD8s were required. And we found that during the primary phase, both were actually contributing to the immune response. So depletion of either CD4s or CD8s completely abrogated our therapeutic effect. However, we were surprised to find that in the memory phase, if we depleted CD8 T cells, the mice actually rejected the tumors just fine. CD8s were not required for rejection of secondary tumors. Instead, we found that CD4s were actually critical for this memory response. So if we depleted our CD4 cells, we completely lost this therapeutic benefit. We went on to check for systemic T cell activation, similar to what we had observed in patients. We found that in mice, looking between day 7 and day 10, especially in the peripheral blood, we saw an increased activation of CD4 T cells in circulation. And you can see here we measured this by both CD44 positive, CD62L low phenotype, as well as by CD11A expression. And you can see right at day 10, we have this peak of T cells in the periphery that diminishes slightly. And we know that that's actually because that's the time point at which the CD4 T cells are actually trafficking to the tumor, which I'm showing over on the right-hand side of the slide. We found that CD40 was critical for this increased systemic T cell activation. And we found this also in the tumor draining lymph node. Immune checkpoint blockade was not contributing at all to the systemic T cell activation, but instead this was solely mediated by agonistic CD40. And I'm not showing it here, but we found that the addition of immune checkpoint blockade was really obvious when we looked in the tumor microenvironment and found that only when you had both immune checkpoint blockade and CD40 together did you see an increased capacity for cytokine production, including cells able to produce multiple different cytokines. Here we're showing that we found CD4 T cells trafficked into the tumor site through the CCL5, CCR5 axis. So we found that if we blocked CCL5 after therapy, we had a significant reduction in the proportion of T cells able to traffic into the tumor site, and that was mediated by CD4 T cells. So if you blocked CCL5, you didn't see any reduction in CD8 T cells. It was just CD4 T cells that were using this pathway. And if we used CD4 T cells that lacked CCR5 expression, they were unable to traffic into the tumor site to the same extent that wild type cells transferred in were able to. So CD40 is activating T cells. Specifically, it's activating CD4 T cells to traffic into the tumor site through CCR5, and that's critical for tumor rejection in this model. So just for some final thoughts here, we found that T cell infiltration into the tumor site is actually crucial for successful immunotherapy. CD4 T cells seem to be the major population that's trafficking in, but we know that CD8 T cells are also required in the memory phase. We found that, in the priming phase. We found that T cell priming requires agonistic CD40. We've been unable to generate really robust T cell responses against this tumor in the absence of agonistic CD40, and that to our surprise, chemotherapy is actually detrimental to this developing immune response against a tumor. This suggests that we need to revisit our sequencing and scheduling of chemotherapy and immunotherapy, not just in pancreatic cancer, but likely in many different tumors. And we think there's opportunity to continue to use chemotherapy and then consider adding in immunotherapy during the maintenance phase or when patients progress or no longer want to be on chemotherapy. And I think that the mouse model will provide a way for us to continue to address this important question. And finally, I think we as a field need to think more about how to harness CD4 T cells in the tumor microenvironment. I think that pancreatic cancer, at least in this system, is giving us a window on the potential of CD4s in being potent tumor rejectors, and we think we have the opportunity to address that question even further in the future. So with that, I would just like to acknowledge my current university, where I have a team that's carrying on with a number of these studies, especially with regard to the CD4 T cell response. And much of this work was actually done at Penn, where I was before I moved to OHSU. The last part of the talk was done by Alex Morrison, who was a master's student in my lab and is now a resident at the University of Pennsylvania. Much of this work was done in collaboration with Bob Vonderheid's group, Ben Stanger's group, also had collaborations with John Ware's lab. And some of the clinical data that I show was done in collaboration with Rosie Mick at Penn, as well as Courtney Betts from Lisa Cousin's lab, with support from Sam from her lab as well. I have my email address listed here, and I'm happy to take any questions over email or through the chat function here as needed. Thank you.